And of course, we're always out there having fun. These are a couple of students I had with me 2 years ago. Beautiful sunny day, and we ran the sunscreen, so we decided to use the mud to see the wolves. So they all came home, and about 2 days later, they all had like white cheeks and red faces. So it's another value for sediment. Okay, final project. We're moving along here. Going along, sorry about that. Wild species and aquaculture interactions. Despite what most of us may think, there is a diversity of wild animals that inhabit the area in aquaculture site. In the tidal, in the shoreline areas next to one, the rocky areas, there's lots of lobsters. On the structures themselves, you'll see fish. And essentially, there's a lot of animals that are living in the area, right? Either taking advantage of the habitat that's being provided by the aquaculture site, or we think taking advantage of the nutrients that are offered by the aquaculture site. And so I'm really interested in looking at, well, what's the interaction of wild species and aquaculture, and what can it tell us? Again, this is, oh, sorry. So this is anemone, just in the soft sediments in your cage site. Now, sometimes species like this are considered a nuisance. This is called biofouling or fouling. This is a close-up picture of the predator net. So it's a net that's been put in place by the aquaculturists to keep out predators. But instantly, or not instantly, but very quickly gets colonized with anything that's floating by, any wild species that's near it. They take advantage of that habitat. And not only do they take advantage, not only do they establish themselves on that, they grow very quickly. So this is a fouling. This is considered a nuisance. To me, it's considered an opportunity.
It's under the cages?
No, this is right, this one would be right on the side of the cages. So these predator nets would be here. This one's probably a little lower. So this is right here, just this would probably be just outside Passamaquoddy Bay. Again, heavily foul predator net, but there's a diversity of species that have inhabited this. Right? You can see the blue mussels. We've got some Siona, some sea daisies, which is a tunicate. But more interesting, if you look really, really close, and the picture picks it inside here. There's all these little tubes here. These are all living animals that have started to make a substrate. They're creating a habitat for other species. This whole process is just like the process of succession in a forest, where we've had deforestation and you would see new species arriving and growing, and they would establish habitat for new species like birds to come in. It's the same thing that's happening on these cage sites, but in a much shorter time frame. This could happen within 6 months. This kind of change. As I was alluding to in the introduction a little bit, the aquaculture industry started to change a little bit in terms of their evolving. They're trying to take advantage of some of the things that biodiversity has to offer. This is a picture of a cage site just outside Passamaquoddy Bay. These large polar circles contain finfish species, those salmon, which are fed. And right along the side of this, there's a number of small polar circles, which they put blue mussels in. And outside this a little further, this is a different kind of raft system where they have algae. And the intention is not only to create products that are diversified for them, to give them some financial stability, but also to see if they can get some environmental benefits by having the mussels extracting the excess nutrients that are coming off the cage site and the same with the algae, the excess nutrients. It's a little bit of an experiment process, but it's a step in a direction that speaks to sustainability and the value of biodiversity. This is inside, we're about 15 feet inside one of those mussel polar circles. And you can see these it's just like a forest of mussels, of blue mussel And they're originally put in socks, little fine mesh socks, little tiny scraps, itty bitty. And in a short period of time, you get these large mussels growing. This is just a close-up. You can see that they're open and actively feeding. So they're obviously not only just taking advantage of the habitat that's being created for them, but they're there to take advantage of the nutrients that are available. Sampling around a cage site is a little tricky. Can't really put a trawl down through there. They don't like that. The benefit grab tends to get hooked up because the environment is quite complex. There's a lot of lines in the area. You can't get everywhere. You can't sample directly from the surface to a lot of spaces. So we tend to do a lot of diving. It gives us a really close look at what's there and allows us to be very precise in what we're looking at. Cameras are one of the simplest and most effective tools that we bring with us. It's just a really simple and accurate way to keep track of what we're seeing down here. So we're often using a lot of cameras. This is just a picture of Dr. Robinson taking a sort of a transect at the bottom, and I think he was taking a picture. Yeah, okay. So he was taking a picture of me. He was adjusting it because my face is good. And that's the picture he's on. So, and the idea, like I said, it really allows us to get up close and personal and to see what's happening to these organisms and how they're responding in their natural environment. So I can see exactly what substrates there are, what species are there, what other species are attached with, and get an understanding of what's happening. Diving, or particularly science diving, is really not as romantic as you think. It's about 50% preparation and planning, and then it's another 50% of preparation and planning. And the reason for that is because not only we're not down there to have fun, we're down there to get a job done. So not only do we have to be safe and keep ourselves, figure out how to keep ourselves dry and warm, keep our zippers done up, but also we're trying to actually achieve something. We're down there collecting information. So it's actually a lot of hard work to get the job done. And so when we finally do get into it, we have to be very efficient with the amount of air we use and what we're down there to do. The cameras help us be efficient. And when we get real close, we can actually see a lot more than what the casual diver would see just passing. This is, again, one of those muscle socks. You can see, this is one of my favorite species. It's so pretty. Silent intestinalis. These fringes fluoresce, and it just constantly filters the water. It'll eat anything between the size of a bacteria, like a micron, to anything that actually fit inside. So a large particles. So it's very, very good food, very efficient, very potent. You can see. And then there's a whole, and then if you look really close, you can't really see here, but if you look really close, there's a whole network of other species that take advantage of this habitat. So how do you quantify this whole succession? How do you quantify diversity in this type of context? And one of the ways we've been doing it is to use essentially a passive collector. So essentially what we do is we introduce habitat, artificial habitat. It's put in clean, so I know there's diversity on it now. And then after about 10 weeks, it looks like that. So it gets all fouled up. But that's the way I can sample diversity around the cage site. Take it out of the water, you can see right away that there's a crust of bryozoans. There's hydrozoans that are growing, these little hair-like things. They're all little animals, hydrozoans. There's a sea vase, there's blue mussels. It's just filled with life, just after only about 10 weeks. And this is responding not only just to the presence of the habitat, but presumably also responding to the nutrients that are available in the area. And if I left that plate there a little longer, I'd expect to get the same kind of succession response, an increase in growth, increase in biomass accumulation, and the same thing I see in the predator net. I have a student working on this through UNB, Jonathan Day, and one of the projects he's looking at is, again, how do you quantify this? How do you, particularly using these plates to see if there's a difference between fouling close to the cage site and fouling far away from the cage site to see if exposure to nutrient levels can be measured. Essentially what he's doing is he's putting these collectors right on the cage site, and this is one of the sites he used, right on the cage site. So he puts these here. He puts another set of collectors about 100 meters away from the cage site, sort of downstream. And then there's actually another set of collectors out here, about 500 meters out in the channel, which is sort of what we consider far away. We'll leave them there for about 10, 15 weeks. And this is the kind of information we're expecting to get. So this is information I got from Huntsman last year on the cage site. British Columbia. Did the same experiment. And I set up two series of collectors, a series of 5 meter depth and a series of 10 meter depth. And they were set at 0 right next to the cage, 100 meters away or 500 meters away. After 10 weeks, the increase in weight of that plate was about 7 grams, 65 grams, 35 grams. Essentially, there was a bit of reduction with distance at that 5 meter depth and the amount of biomass so accumulated.
Do they use every plate at the different height?
Yeah, so essentially I've got a series of replicates. This is sort of an average. These are just error bars on that average. So, and yes, the animals accumulate on every plate that's out there. So it looks like there might be a distance effect with that 5 meter depth. Interesting enough, at the 10 meter depth, the pattern wasn't the same. It's a little less. And the reason for this is maybe that because they're feeding at the surface, that if it's a nutrient effect, if they're feeding at the surface, nutrients may not have a chance to actually trickle down to 10 meters next to the cage. It may take 100 meters for those nutrients to move down. So we're investigating that further to see if there really is a nutrient effect and how wild species response can measure that. So what do we learn from wild species and aquaculture interactions? We've determined what types of wild species colonize an aquaculture site, and that they do this through a process of succession, similar to what we see in the forest. Wild species diversity and growth may help us determine the best placement of habitat from multi-tropic species. Optimal. This is particularly important for the industry that we're trying to put in different species. They have to decide what species they use and how to optimize their growth. So it's good for site design. And in terms of regulatory outcomes or things in terms of regulating these types of aquaculture sites, the wild species diversity and growth may actually help us determine how efficient a cage site is by actually looking at changes in growth over a period of time. So, other sampling tools. Okay, go real quickly here now. This is suction sampling. So I don't know if anybody was curious about this bazooka kind of thing. But this is a very, very simple but effective vacuum underneath the water. And essentially, what this diver is doing is holding it down like this. What we do is we hook it up to an air tank. The air goes in here. Rather than blowing out that way, where we're underwater, air's going to go up, right? So air's going to go up this way. And it creates a suction, just like a draft in your chimney. It'll actually suck up everything that goes underneath. So you're swimming on the bottom, you're hanging onto this thing, it's kind of heavy. And this is my cleaner bag, my vacuum cleaner bag. So all the fine stuff goes out, and anything I want to keep stays in here. So a very, very simple, effective tool, very rugged, really, really efficient use of our time underneath the water. And that's just a picture of me turning it out into you. And I can't apologize for the bad picture, because that is probably as good as it gets down there. Once we get using that thing, there's so much in the water, that's as good as it gets. That's